Hey, ladies and gents, hope you're all doing well today and ready to talk about Chapter 5, looking at big molecules and what they're able to do for us. The molecule you see on the screen right now, this is sort of a top down view of the three-dimensional structure of a protein, particular protein called rhodopsin. Now, sort of taking a step back and thinking about how things are connected, just think that matter, the material of the universe, is made of atoms. And those atoms are held together by all sorts of bonds and interactions. And the biggie is covalent bonds, like we talked about in Chapter 4. In Chapter 4, carbon atoms can form up to four different covalent partners, particularly with other carbon atoms. And as a result of that, you get these really big molecules that have all these different elaborate shapes. And since shape influences function, we wind up with differently shaped molecules that can do all these different jobs. And that comes back to this concept of emergent properties, right? When you put atoms together and make these new, more complex systems, they can do different jobs. And when we get to the macromolecules, these big molecules, when you put them together, all of a sudden you create these structures, cells, that have the properties of life. So life happens when you put together these big molecules. Looking back at rhodopsin, there's another image of rhodopsin from the first slide. And the cool thing about rhodopsin is it is a photopigment that absorbs light energy. That light energy that it absorbs can power different processes. Originally, rhodopsin evolved in soil bacteria, like you see in the bottom right-hand corner of your screen there. Those soil bacteria use rhodopsin to absorb light energy, and then that powers photosynthetic processes for them that helps them make food for themselves. The cool thing about this is that rhodopsin that evolved first in these bacteria, we use in the rods of our eyes to absorb light energy to help us see. So you can take the same molecule, rhodopsin, and it can be adapted in different ways for different processes. All right, so we have four classes that we're going to look at here. We'll do four segments, one for each class of macromolecules. Lipids technically aren't macromolecules because they aren't polymers, but they're big molecules that are really important to the creation of life, so they're always included with this stuff. But pay attention to sort of the atoms that comprise those molecules. You'll find there's a lot of similarity. It's just combinations of atoms and the structures that they perform. So remember, structure is going to influence function, and that's the key to sort of understanding how this variation allows for life to be created. I don't really have a particular reason for this slide. This just used to be in the old textbook, and I thought it was funny because it, looks super serious and high tech, except for the fact that it's like a 35-year-old image. So, pulling it all together, if you just keep big things in mind, everything sort of falls into that context. So you can take really small molecules called monomers and put them together, you form these big polymers, and you can build all these sorts of different structures. So by having these particular building blocks, you can rearrange them in different ways and make all these different things that do different jobs. So the process of tearing down and building up is done through two sets of reactions: dehydration synthesis reactions and hydrolysis reactions. Now, again, the big molecules we make are called polymers. Poly means many, mono means one, right? So you take monomers, smaller molecules, link them together, and you get these big polymers. And that's done through these two sets of reactions. With a dehydration synthesis reaction, this is where you're forming bonds and making larger, more complex structures. What happens is you take a hydrogen from one partner and an OH from another partner, hydroxide from another partner, and join them together, and that forms water. When you do that, it allows a covalent bond to be made, and it pulls, in essence, what can become water or what becomes water out of the separate molecules. That's why it's referred to as a dehydration. And again, it's a synthesis because you're creating that bond. So here, with the smiley faces, you're looking at the peptide bond that's formed between proteins. You pull out an H and an OH, that'll form water, and that forms a covalent bond between the two monomers, between the adjacent molecules. Now, the reverse of this is a hydrolysis reaction. Hydro means water, and lyse means to split. So with a hydrolysis reaction, you can break a bond between monomers by inserting an H and an OH. So when those attach to the individual monomers, it keeps them pulled apart. So what happens is You eat a meal, yum, yum, yum. When you digest that food, it performs a series of hydrolysis reactions. The amylase in your saliva, the enzymes in your stomach, the enzymes in your intestines, they all perform hydrolysis reactions and they cut up your food into its constituent monomers. Then the particle or the molecules are small enough that they can be absorbed into your intestines. They travel through your bloodstream. They get dropped off to the trillions of cells in your body. And then when those monomers are in your cells, then under the direction of your DNA, your cells can build up those monomers into whatever polymers you need them to be, and it becomes part of your body. So when people say you eat, it is absolutely the case. As you sit here, you are an entity that is comprised of the matter that was found in the foods you've eaten over the course of your life. So you take things in, hydrolysis, you break them down, send those monomers to your cells, and then through dehydration synthesis reactions, you're forming those covalent bonds and you're building up the tissues of you. So let's start with our first class of big molecules, carbs. When you think about what carbs do for us, think of sort of two things, or do for organisms. That's an energy molecule, and it's used to sort of build structures within cells. It's an energy molecule because carbs can form rings, and within those rings you have all these carbon-carbon bonds and carbon-hydrogen bonds. And bonds have energy. So just like the sort of teal molecule and then the sort of two-part green molecule that you see there, the black line represents energy, and then the two green dots represent like a disaccharide of sugar. When you break that bond, energy gets released. Okay, so when we consume carbohydrates, you can hydrolyze those molecules and then send that off to other metabolic processes, break those foods down more completely. And when you break those bonds, that powers the creation of other substances that's used for sort of chemical processes of life. Okay, so think about food consuming or food containing energy. Carbs will contain energy. Carbs can also, if you arrange them in a particular way, form these long tendril-like molecules that can interweave and form tough structures like cellulose. So the image you see in the bottom right there includes some of the building materials that could be used with carbohydrates. So when you think of carbs, again, think of them as fuel. Think of them as sugars that can be broken down. Now, they come in different varieties. There are monosaccharides. Those are your monomers. Those are single sugars. Disaccharides, like sucrose, like table sugar, are two monomers linked together. And then polysaccharides can be made with huge numbers, thousands of monomers linked together. Now, knowing that the glycosidic linkages are what connect the monomers of carbohydrates, yeah, I mean, I'm not worried about you like memorizing the specific vocabulary term, but just sort of keep in mind the prefix gly means sugar. So anytime anything is gly, it's telling you, it's indicating to you, hey, they're talking about sugars here. Now, what I do think is helpful is the general formula, the basic formula for carbs. It's CH2O. So you have approximately equal quantities of carbons and oxygens, and you have about twice as many hydrogens as carbons. So keeping that general formula in mind will help you look at diagrams and be able to spot carbohydrates. All right, looking at the notion of how structure influences function, you can cite the example of starch and cellulose. Starch and cellulose are made of the same monomers. They're both made of glucose. But if you arrange the glucose molecules in a different way, if you have them being sort of alternating versus not alternating, it causes it to spiral or stay as a straight molecule. And what that does is allow you to create things like cellulose or starch. Again, made of the same monomer, but used in different ways. Starch, like what you see in these little globules on the left-hand side here, they form what are called amyloplasts. And what they'll do is store this sugar in cells as an energy resource for the cells. Cellulose, on the other hand, the fibers can interweave and form these myofibrils, which are like little ropes of sugar, and they become really tough. And that's the cellulose in the cell walls of plants. So cellulose is sort of a tough almost semi-rigid structure that allows, excuse me, plant cells to have some degree of rigidity. So that's the stringy stuff you get in like celery. So it makes the cell walls much tougher. It makes it difficult for pathogens to get in, and it's a structural feature that provides some support and stability that allows plants to rise up against gravity. Okay, now this is actually a really helpful image to look at because it helps you make sense of identifying carbohydrates. One of the things you'll need to do for the test is be able to look at molecules and know what class of molecule it belongs to. Carbs usually exist in solution, right? Life exists in water. So when carbs are exposed to water, the monomers tend to circle up and form rings. So when you see rings of carbon like that, a lot of times it's indicating a carb. So when you look at it, you see it's made of carbon, hydrogen, and oxygen, right? It's made of CHO, and you see it being in a ring. Another thing I'd point out is the numbering. This isn't super important now, but next semester, the numbering takes on a larger role. If you look, you have what's called an asymmetric carbon. The asymmetric carbon has 4 different partners. So carbon number 1 here has a hydrogen partner, a hydroxide partner, an oxygen partner, and a carbon partner. So it gets the designation as carbon 1. And then from there, you go in a clockwise order. So there's carbon 1, 2, 3, 4, 5, and 6. Okay. So pay attention to the asymmetric carbon. Again, it has 4 different partners, so it serves as sort of like a landmark. And then you number in a clockwise fashion. Oops, I don't know what it looks like for you guys on the screen, like this. So, just pay attention to the numbering of carbons. It's not going to be super specific right now, but again, it will make life a little easier next semester. So again, glucose can be broken down. It's a monomer. The breakdown of glucose is used to power cellular respiration, which is a process of producing ATP in our bodies. So carbs are an important source of energy for us. Okay, now polysaccharides, like starch or in our bodies, glycogen, can store that fuel for long periods of time. So carbs can be sort of an immediate energy source, like glucose in our bloodstream. And then carbs can be stored as long-term energy in things like glycogen in animals or humans and starch in plants. I don't know. I chose that image just because I think it's sort of funny. I can imagine when they try to tell this guy originally, hey, trust me, this picture will turn out cool. He's sort of like, I'm not sure, I guess. Whatever happens, happens. So anyway, you can see all the different ways to use carbs there. So this is sort of an interesting image. You see the differences between the different types of storage carbohydrates or polymers made with carbs. So you can take individual glucose monomers, the same monomer, and arrange it in different ways and get different molecules. First, you see starch. We're all the carbon number 6. You see how the number 6 carbons if we bounce back a couple, here's your number 6 carbon. You can see if they're all arranged in parallel like that, you wind up with something like starch, which would be more of a helical molecule. Cellulose is more of a straight chain molecule. And so starch is energy storage in plants. Cellulose is what interweaves and creates the cell walls in plants. And then glycogen is how we store our carbs. So what happens is if you're an endurance athlete, if you're a distance runner or you swim long distances, you might carb load. So what happens when you carb load is you consume this starch, chomp, 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 amylase in your saliva, amylase in your intestines, cut up the polymer, it cuts up the starch and releases the glucose monomers. And then what we do is we take those glucose monomers and we create these branch chain carbohydrates like what you see in glycogen. So you have those in like little packets of sugar and skeletal muscles around the body, and then you store quite a bit in your liver as well. So when you're an endurance athlete, you consume the starch, break it down into glucose monomers through hydrolysis reactions. And then through dehydration synthesis reactions, you build it back up into a storage molecule that we can use. So then when you have your endurance activity, you can cut up those glycogen molecules, release the glucose into your bloodstream, and have glucose available for ATP production. Gosh, I feel like we've talked about this plenty already. Again, you've got cellulose. It's a structural carbohydrate used in the cell walls of plants. That's why, when sort of thinking of it, if you bite into like a carrot is sort of crisp, right? Because there's water in the big vacuole and it's pushing against that cell wall. So there's pressure on the cell walls. And so when you bite through the cell walls, that's when you get that sort of crisp experience in like an apple or a carrot or whatever the case may be. Okay, now enzymes break down carbs through hydrolysis reactions, right? But enzymes are particular to the type of polysaccharide that they'll be able to break down. So if you take something like cellulose, if we bounce back here, you can see starch and cellulose, they have different, it's the same monomer, but they have different orientations, so they have different molecules. We have an amylase enzyme that can cut up starch. We do not have the enzyme for breaking down cellulose. So that's what I talk about when people say, like, get your fiber. Fiber is cellulose that we can't digest, so it sort of passes through us and scours out your colon, cleans your intestine, that kind of stuff. So fiber is definitely good for you, but we can't digest it. Now, some organisms can, though, because they possess the enzyme needed to break it down. So a cool example is ruminants like cows. Cows will eat grass all day long, right? But cows do not get their nutrition from the grass. The cows feed the grass to bacteria that live in their stomachs, and then the bacteria digest the cellulose, and then the bacteria produce all sorts of nutrients and vitamins for themselves, and then the cows live off of what the bacteria produce. Termites do the same thing. Termites will consume plant material and wood, but termites themselves can't digest those carbs. Bacteria living in the guts of the termites can. So they've experimentally done things where they treat bacteria with antibiotics. They treat termites with antibiotics. And when the termites have their gut bacteria killed, the termites die. So a lot of these herbivores, they have these relationships with microbes that can digest the cellulose. One cool way you can sort of experience how our bodies break down starch is with eating a cracker. If you eat a cracker like a saltine and you let your saliva work on that, it's sort of gross to do, but if you keep a cracker in your mouth for a minute or two and let the saliva work on that, what will happen is the starch will start to get broken down. And when the starch gets broken down into other sugars, it's somewhat sweeter. So a cracker that you chew on for a while becomes sweeter in your mouth because your saliva is actually cutting up the starch into smaller molecules. All right, last carb we'll mention here is chitin. Chitin is what creates the exoskeleton of arthropods. So insects, crustaceans, organisms like that have sort of that flexible shell that is made of a combination of sugars and proteins. So chitin creates this molecule that, again, provides flexible support. Bugfest will be is an opportunity to consume some crunchy snacks because you can eat insects. If you've ever had, fried crickets dipped in chocolate, it's sort of like a Kit Kat bar. So not too bad. All right. Before we move on to the next video, what I'd encourage you to do, just take a moment, pause, and think about some of the things in your life that you're grateful for. I think it's often a good opportunity to pause and reflect in moments and just sort of think about what's good in life and then do that before you move on. Okay? Thanks a lot, guys. We'll see you soon.